Okay. Good evening, everyone. We'll start with the class. It's already going to be 7:15, so to be waiting more. Okay, so today we are going to discuss some questions from the lectures of week 3. Okay, so I hope you all have attended the lecture for week 3 and have joined the class because it is very important for you all to follow the lectures of that particular week before joining this class. Okay, so I'm hoping everyone has gone through the notes and they have come. So, before we start, I want to clear a few things. So, this we are going to have 15 MCQs today, for which we will be given around 1 to 2 minutes for each question. All are very easy and follow the same pattern how you are getting your current assignments. Okay, so this is a practice session so that you all can do your assignments properly. Okay? So we are instructed to be given similar kind of questions compared to your current assignment questions so that you can practice some more MCQs other than the current year assignments. Okay, so I'm not going to teach you again any of the lectures which has been already taught by the professor, or nor am going to give you any notes for revision. This is a practice class where you have to actively participate and answer the questions with me. Okay, try to answer the questions with me. This will test your knowledge, how much knowledge you have gained through the lecture notes, through the lectures, and where you are lacking. If you are unable to answer a question, obviously that concept is not clear to you. You have to go through the lectures again for that particular concept. Okay, so with that aim, we are having these classes so that you can practice and revise the topics again and you can know where you are lacking behind, so you can go back and go through those topics once more. Okay, and also the main thing is so you can do your current year assignments properly, because you carry because those assignments carry marks, right? If you all are, whoever is willing to give exam and get a certificate, need to do the assignments very carefully so that you do not lose marks in the assignment itself. Okay, so we'll start. We have 15 MCQs and I hope everyone knows the rules for the class. Whoever has joined new today, let me introduce myself. I am Amrita, a PhD student at University of Hyderabad, and I will be taking these live sessions every Tuesday 7 to 9 p.m. So you all are requested to join the class maximum by 7:10. 7:15 and I will be starting the class. Okay, and second thing, no need to take any kind of notes. You will be given the class recording as well as the presentations for your reference later. So no need to be in a hurry or no need to take or write down any of the questions shown here. You will be given the PPT. So just focus on answering the questions. So how you have to answer the question? So I will show you the questions one by one and I will give you some time. You think which is the correct answer for that question and write down your choice of answer in the chat box. Okay, again I am repeating, you have to write down your correct answer in the chat box. Please do not depend on others or no need to copy. You can think. If you know, well and good. If you don't know, you are here to learn. You will learn and go. Okay. But just because you don't know the answer, please do not hesitate from answering. Answer it. Whatever you think, either by guess or either by knowledge, answer it. If even if it's incorrect, you will get to know the correct answer anyhow. After every question, we are going to discuss the correct answer as well as its explanation. For some questions, I have kept the little bit detailed explanation. Okay, so yeah, and everyone, please be mute for the audio and for the video throughout the class. Okay, this is the rule to maintain the decorum of the class throughout. Okay, so I hope everyone follows it, and I think I am clear. Please Give me a thumbs up to know that everything is fine and you all understood everything. Okay. Okay. Okay, great. I got thumbs up. So we'll start with the first question of today. Which of the following statements best differentiates enzymatic reactions from microbial fermentations? Enzymes have low substrate specificity, microbes have high specificity. Enzymes require specific pH and temperature, microbes require a broad range. Microbes require defined substrates, enzymes grow in any medium. Both require undefined conditions. Start answering. Whoever has any doubts or any questions, you can write down your queries in the chat box. Okay? I am continuously seeing the chat box. Dhanyashri, I can see you're raising your hand. Can you write down your query in the chat box so that I can know what is the problem? And whoever is present in the class, please try to be active throughout the session. We have 15 questions, and how many of our students are there? Please be active and try to note down how many correct answers and how many wrong answers you all are giving. We will see your marks at the end of the class. This will help you to know uh, at what base you are and how much you need to improve. Okay, so I will need your scores at the end of the session. I need to know how much you got out of 15. So keep noting down how many correct answers and how many wrong answers you all are giving. Okay. Okay, let's see what is the first answer to this first question's answer. It is B. Enzymatic reactions. Enzymes require specific pH and temperature. Microbes require a broad range. Right? This is what differentiates the enzymatic reactions from the microbial fermentations. So the explanation is written here. Enzymatic reactions generally require a specific pH and temperature, whereas microbial fermentations operate with a broader optimum range for growth and metabolism. So the microbial fermentations, there is no such thing like specific pH or specific temperature. You have to test a broader range of pH and temperature to know what is their optimum range for growth and metabolism. So B is the correct answer. So here you can see enzymatic reactions are mostly globular proteins. Enzymes are mostly globular proteins and very specific to the substrate, requires particular pH and temperature, specific substrate to be added. This is what happens in an enzymatic reaction. Whereas in a microbial fermentation, not that much substrate specification for microbial growth. Required pH and temperature would be in the optimum range. Medium to be added, medium containing carbon source for energy, body building and product formation, nitrogen source for body building and product formation, minerals and vitamins as a cofactor. So these are the things required for growth in microbial fermentation, right? Moving on to the next, enzymes lower the activation energy of a reaction by changing the equilibrium constant, altering the free energy change of the reaction, forming enzyme substrate complexes, increasing substrate concentration. Start answering. Okay, I think everyone has answered. Let's see what is the correct answer. It is C. Enzymes. What is the C option? Forming enzyme substrate complexes. Sorry. Enzymes lower the activation energy of a reaction by forming enzyme substrate complexes. Enzymes catalyze reactions by binding substrates to form enzyme substrate complexes, lowering activation energy but without altering delta G or equilibrium constants. Okay, so this is what happens when the activation energy of an enzyme is lowered. When it happens when they are forming enzyme substrate complexes, right? Moving on to the next question. Which type of inhibition cannot be overcome by increasing substrate concentration? Competitive, non-competitive, uncompetitive, allosteric. Start answering. Okay, this is one more thing I want to say you all, that please try to be on time. From next class, I won't admit anyone who is joining after 7:50. Okay, so please try to be on time. If you are unable to join by 7:15, you can go through the recording from the YouTube channel. Okay. Okay, is everyone done? Let's see what is the correct answer. It is B, non-competitive, right? Which type of inhibition cannot be overcome by increasing the substrate concentration? It is non-competitive. In non-competitive inhibition, the inhibitor binds to a site other than the active site, affecting both the free enzyme and the enzyme substrate complex, making increased substrate ineffective in overcoming inhibition. That is why you can't overcome the inhibition by increasing the substrate concentration in non-competitive type of inhibition. So we'll see here, this is from the slide. The rate equation of enzymatic reaction with non-competitive inhibition is given here. I hope you all have gone through this. So it is already written here, non-competitive inhibition cannot be overcome by increasing the substrate concentration. Moving on to the next. In uncompetitive inhibition, the inhibitor binds only to free enzyme, only to enzyme substrate complex, to both free enzyme and enzyme substrate complex, to the substrate. Start answering. Okay, I think everyone's done. Let's see. The correct answer is B. Only to enzyme substrate complex. So in uncompetitive inhibition, the inhibitor binds only to the enzyme substrate complex and increases the substrate concentration. Increasing the substrate concentration actually favors inhibition, right? So here you can see uncompetitive inhibition. This is from the slide. Inhibitors do not bind to free enzyme but affect enzyme reaction by binding to the enzyme substrate complex at a location away from the active site. Here you can see. So the next this one I have kept so that you all can see it once more because these things are important. Competitive, non-competitive and uncompetitive. You should not get confused. So what happens in the competitive reaction? The inhibitor binds to the free enzyme only and compete with the substrate. This is called competitive inhibition because it is competing with the substrate. Increasing the substrate concentration can overcome the inhibition, right? Next is non-competitive inhibition. The inhibitor binds to the free enzyme or the enzyme substrate complex. It is not competing with the substrate for that same site. It is binding to a different site and it can bind either to the free enzyme or to the enzyme substrate complex as well. That's why it is called non-competitive. Increasing the substrate cannot overcome inhibition. So here, if you even if you increase the substrate concentration, it cannot overcome inhibition. And the third one is uncompetitive inhibition, in which the inhibitor binds to the enzyme substrate complex only, favoring substrate and increasing the substrate concentration favors the inhibition. Okay, so here the difference between uncompetitive and non-competitive is the substrate concentration. In non-competitive, increasing the substrate cannot overcome. Uncompetitive, if you increase the substrate, it can overcome. Okay, so moving on to the next question. In plug flow reactor, there is complete axial mixing. There is no axial mixing, only radial mixing. Fluid elements interact continuously. Composition is constant throughout the reactor. Okay, so there is a mistake in the slide, I guess. Uncompetitive inhibition also cannot be overcome by substrate concentration. Right? We already discussed, but in the slide it is written wrong. So you please cross check with that. Okay? I guess only one person is listening to the class properly and able to point out the mistakes. I don't know what others are doing. Why anyone else could not point that out? Already we discussed a question based on that, right? And you all are going through the lectures and lecture notes. You all have opted for this class, then you should be thorough with it, right? These kind of things should not confuse you. You should be attentive. Okay, let's see what is the answer to this question. Answer is B. There is no axial mixing, only radial mixing. So the plug flow reactor with discrete plugs of liquid moving without axial mixing, though radial mixing ensures uniformity across the cross section. So here you can see the plug flow reactor is an alternative to the CSTR for continuous operation. We have discussed about the CSTR in the previous class, right? Right? Liquid entering the reactor passes through a discrete plug and does not interact with the neighboring fluid element. There is no axial mixing, only radial mixing. Composition varies along with the flow path, right? Moving on to the next. One of the first industrial uses of immobilized enzymes in Japan was production of antibiotics, production of L-amino acids using amino acylase, conversion of glucose to ethanol, synthesis of vitamins. Whoever is raising their hand, they can write down the query in the chat box. Okay, let's see what is the correct answer. It is B. Production of L-amino acids using aminosilase. This is one of the first industrial uses of immobilized enzymes in Japan. So in 1969, immobilized amino acylase from Aspergillus oryzae was used for industrial L-amino acid production. Right? Here you can see it was given in the slides as well. I am not reading it again. Which is not an advantage of immobilized enzymes. Reusability, increased stability, enzyme-free products, always increased reaction rate. Start answering. Okay, let's see what is the correct answer. It is D. Always increased always increased reaction rate. So this is not an advantage of the immobilized enzymes. Immobilizations offers many benefits like reusability, stability and product purity, but the reaction rate may not always increase. It can even decrease due to mass transfer limitations, right? So there is not always a case that it will be increased reaction rate. So here are this is what it is written here. Immobilization is defined as the localization of cell or enzyme in a distinct support or matrix. The support or matrix on which the enzymes are immobilized allows the exchange of medium containing substrate or effector or inhibitor molecules. Immobilization enhances the stability of the enzyme. And these are the listed advantages of immobilized enzyme. Okay, so here you can see that there is no advantage where it is written that they can have increased reaction rate, right? So that is the correct answer. Moving on to the next, a good immobilization matrix should be expensive and disposable, inert, strong, and possibly regenerable, reactive with enzyme active sites, poorly available to the substrate. Start answering. Okay, is everyone done? Let's see what is the correct answer. It is B. Inert, strong, and possibly regenerable. This is what a good immobilization matrix should be so that it can be supportive. Ideal supports are inert, physically strong, stable, and economical, large available surface area. Right? Moving on to the next. In covalent binding immobilization, which reagent is commonly used? Sodium chloride, glutaraldehyde, ammonium sulfate, magnesium chloride. Okay, let's see what is the correct answer. It is B. Glutaraldehyde is a common polyfunctional reagent used in cross-linking enzymes without a support matrix. Okay, moving on to the next. Which factor does not directly influence immobilized enzyme activity? Initial substrate concentration, concentration of immobilized enzyme, carrier dimensions, enzyme molecular weight. Start answering. Okay, let's see what is the correct answer. It is D. Factors such as substrate concentration, enzyme loading, temperature, pH, and carrier dimensions affect activity, but molecular weight of the enzyme does not directly influence immobilized activity. Right? We all know that the enzyme molecular weight does not affect the enzyme activity. Okay, moving on to the next question. External mass transfer resistance occurs during diffusion within the carrier particle, transfer from bulk liquid to the enzyme surface, reaction between enzyme and substrate, enzyme desorption. Okay, let's see what is the correct answer. It is B. Transfer from bulk liquid to the enzyme surface. So, external mass transfer resistance includes transfer from the bulk liquid to the unmixed liquid layer around the enzyme and the diffusion through that layer. So, this is what occurs with the external mass transfer resistance. Moving on to the next, intraparticle mass transfer resistance is due to slow bulk fluid movement, diffusion from carrier surface to enzyme active sites inside the particle, denaturation of enzyme, competitive inhibition. Okay, let's see what is the correct answer. It is B. Diffusion from carrier surface to enzyme active sites inside the particle. Intraparticle resistance is the limitation of substrate diffusion inside the support matrix to the active site of the immobilized enzyme. Moving on to the next question. Which microbial growth phase is characterized by metabolic adaptation without cell division? Lag phase, log phase, stationary phase, death phase. Start answering. Okay, let's see what is the correct answer. It is A. Lag phase. In the lag phase, cells acclimatize to new conditions, synthesizing enzymes and preparing for division, but not yet multiplying. So, multiplication generally happens in the log phase, but in the lag phase, the cells acclimatize to the new media conditions and then start synthesizing enzymes and prepare for the division, but do not divide, right? Here you can see the microbial growth cycle. So this is the lag phase. then is the growth phase, which is the log phase, then is the stationary phase in which the growth becomes stagnant, right? And last is the death phase in which the growth starts decreasing if there is not enough media for growth anymore. So here you can see the description as well. Lag, cells adapt to the new environment, no or very little growth. Acceleration, growth starts. Growth achieves its maximum rate. Decline, growth slows due to nutrient exhaustion or buildup of the inhibitory products. Stationary, growth ceases. Death, cells lose viability and dies. Right? Moving on to the next question. In a monode chemostat model, the dilution rate is ratio of substrate consumed to biomass produced, inverse of residence time, proportion to yield coefficient equal to mu max at steady rate. Please keep yourselves mute throughout the class. Okay, let's see what is the correct answer. It is B. Dilution rate D is defined as the inverse of the residence time in continuous culture systems like chemostats. Right? So, next question. At washout in a chemostat, mu is less than D, mu is equal to mu max, D is greater than mu max, mu is equal to D is equal to mu max. Start answering. Okay, let's see what is the correct answer. It is C. Washout occurs when the dilution rate exceeds the maximum specific growth rate, that is mu max, causing biomass concentration to drop to zero. Right? So C is the correct answer. That D is greater than mu max in a at washout in a chemostat. Okay, so this was the last question for today. So let's see what are your scores out of 15. Whoever wants to share, they can write down their scores in the chat box. Okay, only one person has answered. 12 out of 15, 10 out of 15, 13 out of 15. Okay, okay, so that is fine. So whoever has not given, like whatever mistakes you all have done, 14 out of 15, very good. OD doesn't decrease. It's either stays the same. It doesn't decrease. Okay. So if you have done a culture, then you will know that OD doesn't decrease, but after a certain point of time, it stays at the same point. Okay. So I could see a few people giving their scores, and yeah. Is there any more questions, or we can disperse? And I request everyone that next class, please try to join by 7:15, or else I won't be admitting anyone after that. I think I have to make it a strict rule so that you all can be on time. And whoever tries to join after 7:15, I won't admit them. They can go and see the YouTube recording. Okay. Yeah, about the sums, I think I won't be doing it because I have been instructed that I should follow the same pattern with which you are being given the current year assignments. So I went through the current year assignments, and I don't think you all are being given any kind of sums in that, right? So, yeah, that you have to practice from the lecture videos. Whatever lecture videos are there, you have to practice it from there. So here I am only discussing the multiple choice questions which you all will be getting. Of course, you will be getting subjective questions, and in subjective questions, you can have the numericals. But here I am only assigned to practice, make you all practice the multiple choice questions. So that's what I'm doing. Okay. If you have any doubt in the multiple choice questions we have discussed here, or what if you have any doubts in the current year assignment questions you are getting, then I can clear it. But other than that, I don't think it is possible. Yeah, you have to, you have the discussion forum. You can write down your queries in the discussion forum that if you are not unable to understand any of the numericals in the lectures, pat like no one I have not been discussed that or no none of the NPTEL team has told me what will be the pattern of the exam. So yeah, I'm not sure, but I can say that there are going to be MCQs plus subjective questions. It won't be only MCQs or it won't be only subjective. It will be a mix of both. Okay, but exactly how many marks MCQs and how many marks uh, subjective will be there, that exactly I don't know, but I think it is some details are written in the NPTEL website. You can go and check them. Okay. Okay, let me explain once more the competitive and uncompetitive part, but that slide is wrong, as someone pointed out. The theory is written wrong there. I'll correct it. Let's see. So here, competitive inhibition. What is competitive inhibition? That there is only one active site in the enzyme and both the substrate and the inhibitor are competing for it, right? Both wants to try to the bind to the same site. So they are uh, competitively, the inhibitor competitively tries to bind to the free enzyme only and competes with the substrate. So if you increase the substrate concentration over here, then the inhibitor won't be able to bind to the free enzyme and it can overcome inhibition. So this is what is competitive inhibition. What is non-competitive inhibition? In non-competitive inhibition, inhibitor binds to the free enzyme or the enzyme substrate complex because there is no competition here. In this, what is there? In the free enzyme, there are two sites. One is for inhibitor and one is for the substrate. So, increasing the substrate cannot overcome inhibition. So here, as there are two sites, there is no competition. Even if you increase the substrate concentration, it cannot overcome the inhibition. Similar thing happens in the uncompetitive inhibition where inhibitor binds to the enzyme substrate complex but not to the free enzyme. It only binds to the enzyme substrate complex. But here it is written increasing the substrate concentration favors the inhibition, but that is not correct. Here also if you increase the substrate concentration, it won't remove the inhibition or it won't overcome the inhibition. So here it is written increasing the substrate favors the inhibition. Okay, so that is correct. It won't overcome inhibition. If you are increasing the substrate, it won't overcome inhibition, but it will favor the inhibition. So this is written correct only. I was thinking it is written overcome can overcome inhibition, but no, it is written favors the inhibition, so that is correct. What about assignment? Which assignment? Which assignment? Pasna. Are you like what do you want to ask? I'm not clear. Can you say a little bit in detail? Every week, the live class is a Tuesday 7 to 9 p.m. I hope you all are clear with this now. Already we have passed 3 weeks. And I think you all are getting reminder mails as well every Monday, right? So there should not be any doubt with the timings and the days. Okay, I guess this is it. We'll stop the session here. We'll uh, join again next Tuesday at 7 p.m. As I told, I won't be allowing anyone after 7:15. So please be careful of the time. 12. Okay, Upasana, I'm still unable to get what you are trying to say. If you can be a little quick, I can answer your query or we can disperse here. Okay, so this is it for today. Thank you, everyone.